An article I stumbled across on scienceandnonduality.com made my eyes pop out. Two scientists leading an experiment at the University of Washington in Seattle found that the hour of our death is not the hour of our death for the whole of our physical body. According to new research, hundreds of our genes live on or even come to life for the first time for as long as 48 hours after the declaration of clinical death. So one of these scientists, Peter Noble, is on Skype with us. Peter, what happens to the cells when we die?
Well, we looked at the fish and mice. We killed them and then studied what happens to the genes and how they are expressed from the time that you die to 48 to 96 hours. We did that because we were just interested to understand what happens to the genes. We thought, for example, that the genes would just be turned off or decrease. But what happened is that in some cases, like 1% of them, they increase. And we were totally shocked by that. We didn't plan on this in the study. But what is so interesting is the types of genes that are turned on. For example, several of the genes are involved in embryonic development. And the question is, why would genes involved in embryonic development, which are only turned on at the very beginning of life, be turned on when we die?
What's your opinion?
I beg your pardon?
What is your feeling? What is your opinion? Or are you not allowed to talk as a scientist about what you feel about something?
No, I can talk about it.
Yeah?
I have a hypothesis that what happens is the nucleosomes which wrap the DNA are becoming unwind when you die. And those previously silenced genes, because you don't have embryonic genes generally turned on in life. So what happens is as they're unwrapping, then expression is occurring. And as this expression is occurring, that is what we are seeing in our study. More research has to be done on this. But it's sort of interesting to see. We didn't expect this result.
It's so fascinating. It almost ties in with the Eastern philosophies of as one dies, one's getting ready for a rebirth. I mean, it's not too far to say that. It seems very unusual that these genes that are to do with fetal development shut off once we're born, generally. And then here we are in 24 to 48 hours after death, and they are activating, aren't they? That's what you were seeing in the mice and the fish that you studied.
Yes, very much so. And another interesting fact that goes along with this is that I just serendipitously screened the genes that were upregulated or turned on, and we found that many of them are associated with cancer.
Yeah, I read that one.
Why this is important is because as time goes by, since after we're dead, as the arrow of time moves forward, what happens is that there's more cancer genes turned on. Now, why is this relevant? Well, it's relevant because if we look at organ transplant, for example, liver transplant, as soon as you take it out of the host that's going into the recipient, well, there seems to be a time plays a very important role here because it could be turning on cancer genes. We don't know. We do know that it increases, but more research has to be done to find out if this is actually true and it occurs in humans. Remember that we did this study in zebrafish and mouse.
So I'm not quite following you there. Are you saying there is a risk that cancer genes are being turned on in those who there could be a risk where there's some organ transplant?
Yes, what happens, when a person dies from a motorcycle accident, for example, and he's a liver donor, well, there's a certain period of time that liver is in the host. Well, what could happen is that the genes are expressed, cancer genes are expressed, and as time goes by, more and more cancer genes are being expressed. So what happens if that liver goes into recipient where those cancer genes are turned on? And if you actually look at the literature, you find that people who get liver transplants are have a higher incidence of cancer than those that do not.
Wow. That is fascinating. I loved another part of the article where you were talking about post-death. DNA normally has quite a chaotic patterning as it's dying off. But in these, you call them messenger RNA genes, these mRNA genes that activate post-death, they're quite ordered, aren't they? They have peaks and troughs, and they seem to be doing their thing quite independently of the supposedly dead person.
This was the most interesting thing. Like, actually, no one wanted to work on this study. They did the study in 2011, and I just looked at the data and saw the pattern and was just amazed, because I didn't expect. I thought it would be random, but no, there's an ordered pattern, and that was a surprise. And then I said, oh, maybe it's just in the zebrafish. But then I did it in the mouse and found another ordered pattern.
It's a fascinating rabbit hole you go down. Do you feel excited about your work every day? Do you look forward to what you're going to discover in this new frontier?
Oh, yes, I am. I'm very excited. Yeah, I just have to get new funding.
I think we are too, and we'd love to follow your work. Thank you so much for talking to us today.
Oh, thank you. It was exciting.
Fascinating. Thank you, Peter.